Okay, so I will continue on vaccine consideration and mainly talk about Leishmania antigens and how you can bring your favorite antigen to the clinic. What are the bottlenecks and what you have to do. And so first, I want to show my lab is here, okay? In the vineyards. It's not true. in the middle of the vineyards. Okay, so we can classify simply 3 types of vaccine. You have live vaccines, including attenuated bacteria or virally based vaccine, kill pathogens or subfractions, or molecular, very well-defined vaccine like synthetic peptides, recombinant DNA vaccine, virus-like particles, VLPs. So ideally, I think, first thing, it's the efficacy with a long-term protective immunity. That would be your ideal vaccine. Then it must be safe. It must be reproducible from batch to batch. Low cost because it's a neglected disease. So if you come with an anti-L10, great, but I think it will be quite difficult to sell or to deliver in the countries. Large scale, so it means you need to produce at large level, and when have an approved system of production. And I think one point also which is quite important, it's how many, the compliance, people will accept this vaccine, implementation, how many doses, and what are the time between the different shots that you will probably use. For a vaccine, what you need to do is first, I think you need to define your material. And everything needs to be defined. If you want to produce a recombinant vaccine, either in, for example, in eukaryotic cells or higher eukaryotic cells, for example, or yeast, every single component must be defined, and you need to have a proof of the quality of your product. For example, if you use a serum or any kind of amino acids, for example, they need to be defined. You need a sheet showing the quality of your amino acids. So you need certified producer, and you need a consistency, so quality control of the product. You need SOPs. So for each step, you need to really define the procedure of operation. So how do you do things? I take exactly, you have to write down and certify exactly what you did. Quality assurance, so it means people need to be trained. So quality assurance people, quality control people. You need to define, to be sure on your equipment. It goes from the balance to the pipette to everything, okay? So to be sure that at the end you have a batch-to-batch reproducibility in a quality control and assurance environment. And I think this is a key. So really, if you want to go ahead with your antigens or any kind of system and you really push it, you need assistance to do that. Probably there are a lot of CROs, clinical companies who can help you to start to develop and set up your system. For live or kill vaccine, you require less in terms of operating procedures. But what the problem you could have is the culture of your parasite, for example. And it's not trivial. And also defining a definition of a sera. Can you avoid serum, for example, and use synthetic compounds? So can we generate a vaccine? I think probably everybody here thinks it's possible. We know that a spontaneous or drug-induced recovery from leishmaniasis is accompanied by solid immunity against reinfection. But we know that that's a case of leishmanization where people have been injected. Sometimes, unfortunately, these people develop lesions. Then they have to be treated with drugs. So but it means a vaccine is possible at least for cutaneous leishmaniasis. The principle behind is what Paul said, comes from this old experiment of Th1 and Th2. Everything is based on that. So Th1 is good, Th2 is bad. And it comes from the mouse model with a non-healing BALB/C model where you infect with, for example, here with Leishmania major, you develop lesions in the foot pad, you know that you have a Th2 response. If you take a non-healing BALB/C mice infected with Leishmania major, and what you can do, you can treat with interferon gamma or IL-12 or anti-IL-4. you develop a Th1 response, and you can protect against the infection. Okay, that's a Th1, Th2 paradigm. And the Th1 cells have been, I mean, the beginning of these studies. If you can see, it's now 20 years of this first experiment and the first detail of Th1, Th2 balance, where you can, with Th1 cells or specific T cell, Leishmania T cells can be transferred to a biopsy mice which have been irradiated. So you transfer the cells, then you infect, and what you can see that you generate resistance. So everything is based on that, on a mouse model. So one of the problems which was raised also by Paul is the fact that we think that you have presentation of the peptide, and you generate Th1 cells, which produce interferon gamma. Interferon gamma induce INOS, and INOS is responsible for the killing. But that's really the case in the mouse. In humans, it could be hypochloric acid, which is made, and probably the role of NO is not so clear. However, what is probably important is to generate an oxidative burst to kill the parasite. So in fact, you play between Th1 and Th2 and the macrophage, the infected macrophage or DC. And the macrophage can induce and generate Th1 cells or Th2 cells, depending on the type of interleukin or cytokines which are produced. What is important is to see that if you have a Th2 response, it will produce IL-4, which will block the generation of Th1 cells. Th1 cells, on the contrary, will produce interferon gamma, which will block the differentiation of Th0 cells to Th2. So the rationale for developing a vaccine is we know that Th1 cells are essential for resistance, so CD4 Th1 cells. CD8 T cells are involved in memory, and Treg cells are involved in persistence of Leishmania infection. So it means that you need to play to favor Th1 cells and to downregulate Treg. So, and probably one of the problems with the production by the presence of Treg cells is the production of interleukin IL. IL-10 is an anti-inflammatory cytokine. So it's derived from different sources, as Paul mentioned, DC, macrophage, B cell, and so on. And it's known that IL-10 knockout mice are highly resistant to Leishmania. So IL-10 inhibits vaccine efficiency. They are probably in the case of vaccine, they could be produced by Treg. So you have to control the Treg to avoid the persistence of Leishmania. But maybe it's important to keep some parasite inside. And so probably it comes to one ratio. It's a level of IL-10 versus a level of gamma interferon. What is important, it's a balance between these two molecules. So in humans, it's known there is a correlation between the Th1 response and resistance and healing and CL. The protection requires Th1 cells response with IL-12 and gamma interferon production, but low IL-10. That's probably the key elements. So now, if we look at a different type of vaccine and the problem which can be raised. Human live vaccines, the first generation, Leishmanization, so it's a cowpox principle, as was said before, and it was used in Uzbekistan. The problem is mainly the persistence of the parasite. And you have problem with susceptible individuals, immunocompromised individuals. You could imagine that probably using specific replicative deficient parasite on the long term could be a way to go. You could also imagine that you can add specific adjuvant to it, or you can treat your parasite with UV. And there are different ways that probably should be revisited these days. Because when you see recombinant vaccine, it's still not easy to go for it. Anyway, the other possibility is all killed parasite. I would say I had the opportunity to test some of these killed parasites, and I was asked to look for what do you have in the tube? ALM, for example, somebody, I don't want to mention the name, gave me a tube of ALM and said, can you check what you have in the tube? In the tube, there was nothing left. okay, except lipids and a few amino acids. And that's, so I would be extremely careful. It depends how you kill vaccine. Because if you you kill your parasite and just leave it on the bench, GP63, which was mentioned before, will wipe out everything. So you could have maybe GP63 plus lipid plus amino acid and small peptide. That's it. So I think probably one of the reasons of these problem with this kill vaccine, it's the way you prepare them. And so what was left in the case I analyzed was only LPG, in fact, which is quite interesting because it's a toll-like receptor 2 activator. But the really kill vaccine really failed. And maybe they could be interesting to be using with drugs. I mean, that's probably one of the way which is going to take more and more importance is to treat people with antimony and come back with any kind of kill vaccine or recombinant like a therapeutic vaccine. And though it's more, you're acting more as an immunomodulator as was mentioned before. Second generation, I just mentioned some. You had the fraction, the Leishmune, the Canilish, and so on. These are fractions, more or less defined. In the case of the Leishmune, which is a fucomanose ligand associated with quilay. The other one with the one of Virbac, where you have an association with a specific adjuvant, which is MDP. And the one of Steve Reed, I think, which is quite interesting. Even if it failed, I think it gives something a way to go to vaccine development. And it can always, what people do, normally, if you look at the way people think of vaccine, the first thing they do, I'm going to generate a prophylactic vaccine, okay? They do the trial, it doesn't work. Then they go for therapeutic vaccine, doesn't work so well. So then they say, okay, now I take a drug, and I go with my vaccine. So I think it's quite interesting because people try to save somehow the vaccine, which is fair, which is okay. Because at least I think this way you have some molecules that you can test and which can be used as a baseline and see it's better than what existed before, okay? But I think it's quite interesting the way, but probably mixing drugs plus immunomodulators or Leishmania antigens could be quite interesting. And I come back to what Paul said, and I think it's quite important that we have a series of antigens which have been tested, okay? And I don't want to, if somebody here is working with a vaccine as a nice antigen, maybe switch to something which already exists and try to push it, because some of them have already been pushed to different model system. And what can we do to improve them, probably? We should go back probably to some antigens which already do exist. Then, one point with a defined vaccine of FMS is fucomanose ligand. I think this Leishmune, I think there is one thing which is quite interesting in this vaccine, even it was for dogs, was the reduction of the transmission. And then globally, I think that's quite important. You can have maybe something which was working a certain percentage in dogs, but I think the reduction of the disease due to lowering the reservoir is maybe something which is quite important. So maybe you have something which is not 100%, it will never be anyway, works to a certain level, but probably you decrease the transmission of the parasite. As said, there is a series of antigens which have been tested. Most of them have been tested in mice, and only a few of them have been tested in monkeys and dogs. That's one of the problems. One of the key issues is really the financial support you can get to do even your dog trial. It's not cheap. And so some years ago with Paul, we spent quite a lot of money on dogs. Now, if you want to develop your vaccine, what are the questions you have? Okay, the first one, against which species? Don't think it's going to be the same for CL, MCL, or VL. Are you going to go for prophylactic or therapeutic? Which kind of animal models are you going to use? Mice, hamsters, monkeys, dogs? which is the target population, children, adults, elderly. You should know that the immune response of kids is different from adults, which is different from elderly people. So that's what is your target population. The antigens, purified DNA, attenuated vaccine, virally expressed bacteria. Which kind of adjuvant are you going to use? The protocol of immunization, the challenge, experimental, natural, and which are the parameters of, you have to define the parameters of production. So what you have to do clearly is to say, okay, I want to go to humans. What are the conditions to go to humans? You define the conditions. And starting, you go backwards. So, okay, I want to develop a vaccine for humans, so I better be careful with my SOPs, with the production, and so on. So if we take, for example, the antigens, whole parasite can be live, dead, attenuated. There are problems if you try, for example, dissemination of genetically modified organisms. That's could be one key issue. You can have antibiotic-resistant gene in your Leishmania parasite, for example. You can have horizontal gene transfer. Is it possible? You have sex in Leishmania. So depending of if you use all parasite, if they are alive, as if they are alive, or attenuated, you can have a series of problems that you could face. Purified Leishmania antigens, recombinant, I think that can be quite clean. Peptides, pretty clean. DNA, pretty clean. Then the problem you could have is you cannot mix different processes. So you cannot really take something which is, for example, peptide with DNA and mix it and go into humans. That's a problem. Okay. So you should know that if you test your vaccine in a mouse, you better not mix things. Okay. Say, okay, I take a recombinant protein and I go with a specific adjuvant. So, and what you need is quality control of your antigens and reproducibility. So for example, if you want to say, okay, I think probably recombinant antigen is great. What should I do? The first thing you have to think is use an approved system of expression. So E. coli, bacteria, VLPs, yeast, viruses, which have been tested before. Okay, don't go for a fancy new expression system because you will never go through regulatory affairs. Okay, that's what happened to me years ago. QC and reproducibility, the yield, purity, potency, stability, toxicity, immunogenicity. That's what you have to do. You should know your antigens. So it means you have to make QC analysis. So for example, for a recombinant, SDS gel in reduced versus non-reduced condition, Kumasi staining, Western blot, size exclusion. Do you have aggregates or not? You can have aggregates. Doesn't matter. It's not a problem. The problem is, are these aggregates reproducible? Okay, if you can reproduce your aggregation, it's fine. But if you cannot, if you have, let's say, sometimes 50% of the protein which is aggregated, sometimes you have 10%, forget it. And we did some trials, in fact, for malaria with aggregated CS protein. Worked fine, but we knew it was a CS peptide. It was aggregating, but we knew exactly how to do it, and we always had the same percentage of CS peptide aggregated. We went into humans, trial failed, that's okay, but we had something which was very clean. Okay. So there are a series of things you have to do, and that depends very much if it's a recombinant or something else. So N-terminal sequencing, MS, sterility, stability, endotoxin, amino acid composition analysis. So I put here, I think there is a paper by Kamesipo in 2006. We summarize a bit, and you have all the detailed peptide map, oxidation, level of your protein, carbohydrate analysis, N-terminal sequencing, biological potency, purity, SDS, size exclusion, and things which are quite important, which are host cell protein. What is the percentage of the protein of the cell system which was used? For example, you must be under 500 nanogram per ml. Endotoxin level, DNA content, for example, if it's a protein, how much DNA is left, and bioburden, is it contaminated with bacteria? And I think if sometimes we have a lot of series of nice publication in vaccine or in many different journals, and people come with a recombinant antigen in E. coli, and they say there is no endotoxin, but they don't show the data. So you really need to know how much endotoxin you have, which can really affect the immune response, and also the DNA content. For example, here, this is one antigen that we produced. It's a Kumasi. This one was pretty nice, but it's a Kumasi. So if Kumasi staining, but if you look behind, you can still see some band of the, okay. But this one was nice enough. Then we went for HPLC. You see the quality of, no, we don't have any, that was a front. We don't have any contaminants. If you could have a contaminant, that's not a problem, if it's always here, okay? That was one of the problems of many years ago when people started to develop vaccine against malaria and was rushed to develop a vaccine, trying to develop a vaccine against using the CS. But they realized that there's a small contaminant that probably they never figure out what was the contaminant in this CS prep. And because of that, they stop all the vaccine program. So you have to be careful what you do. So you better define exactly what you have. If you have a contaminant, you have to try. But now we have tools to define exactly what it is. Before we had not. So here you have MS, the spectrum. Here this is the molecular weight of our protein. It was pretty clean. Now which adjuvant? Which is another question. I think it's a very important question. So what means adjuvant? Adjuvant comes from the Latin adjuvare, which means help. So they are defined by the real results they produce. So result expected, they must improve the immune response, overcome impaired immunogenicity, hopefully reducing antigen dose, that can help, and outwitting the disease. So, and that they are extremely, I think, probably important, extremely important for weakly immunogenic antigens. So how do they work? You have two types of adjuvants. You have the one which are what is called delivery system, and some which are immunomodulators. And so delivery system, they can improve the immunoavailability, and the immunomodulators, they improve working as co-stimulatory. So they can be, and we will talk about that, about PAMs and DAMs, and the examples are for delivery system. It's a mineral salt, emulsion, or particulates, and immunomodulators. You can have TLR agonists, which are coming, saponin, or you could imagine cytokines. So if you look at the different type of adjuvants, mineral salt, emulsion, TLR agonist, particulate, or others, if you look out of all these adjuvants, the one which are licensed for human use, you have only a few, mainly alum, MF59, which is an oil in water, AXO3, which is oil in water plus tocopherol. And then you have MPL, which is activate, it's like an LPS-like. It's a fraction of, it's a muramyl dipeptide. I never remember. ASO4 and virosol. These are the one which are these days licensed to be used in humans. I think it's quite interesting to look at the history of adjuvants. If you look, alum, it's a very old adjuvant, okay? It comes in the 1930, until 1990, nothing else. It really came with MF59 for the flu. I'm saying used in human. I don't say new adjuvant that people are trying in the lab. So license and adjuvant in human, MF59, liposomes, then started ASO4, ASO3, and that's mainly due, for example, due to pandemic flu, where people are trying to lower the dose or to lower lower the dose, for example, and increase immunogenicity. I think it's also interesting that the mechanism of alum was described only in 2007 or 8. I don't remember exactly. Why we have only so few adjuvants, it's a question of safety, manufacturing, and cost, and most of the time we don't know the mode of action. So normally an adjuvant is not licensed alone, it's always it's always licensed in a vaccine. Okay, it's part of a vaccine. And one of the problems is many of these adjuvants were proprietary of pharmas, and it was very difficult to get. So among the major one, mineral salt, so aluminum in different formulation, like aluminum hydroxyphosphate and calcium phosphate and so on. Oil in water emulsions, which is coming since 1997, which mainly due to influenza infection. And new adjuvants have been licensed, like MPL, which is a TLR4 agonist. So which are the ones which are in development? Now you have a whole series. Okay, people realize that we need adjuvants, and most of them are the ones which act through TLRs, 3, 4, and so on, saponin, polysaccharide, and so on. They are developed because they are mainly either for cancer, vaccination, or malaria, or HIV, HBV, and so on. So you can see the development of these different adjuvants. So I think it looks better than some years ago with the development of clinical trials on this adjuvant with specific recombinant protein, or virosome, or VLPs, and so on. So the problem you could have with adjuvant is hypersensitivity, induction of autoimmunity. You can have fever, local intolerance. I think that you always know sometimes when you have been vaccinated and you have, it hurts, irritation, pain, redness. This is due to inflammation. Systemic toxicity, reproduction toxicity, genotoxicity, and everything which can relate to a specific person. There is no relation between adjuvant and autism, okay, and vaccination and autism, and alum and specific disease, okay? I think that we should really stress the point. If you have to choose an adjuvant, what should you do? I think first, if you can use one which has been used in an approved vaccine, that's the best. Then take a generic form, or use an adjuvant which has exactly the same mechanism of action. If you want to use, if you know the mechanism of action, for example, alum, and you could imagine to use another adjuvant, you cannot have access to that. You say, okay, I know this adjuvant will work the same way than alum, for example. Or use something which is in clinical trials, for example, one of them. That's one possibility. And these guys are pretty clean, okay? So you don't need, and you can have certified documents for these different molecules. Then, if you cannot answer this question, if you don't get anything from these first 4 conditions, you can use an adjuvant with extensive preclinical trial, mode of action to be the same in mice and men. It's not easy to define. And if it's not available or suitable, I think probably think carefully. I would not do the experiment. Okay. So clearly, adjuvants are clearly important for modern vaccine. By default, do not use an adjuvant. If you are here, try to avoid it. It's going to be a problem. Many licensed vaccines contain mineral salt. Very few licensed vaccines contain modern adjuvants. Most of them are oil in water and TLR-based these days. Safety is a key issue, and you can always go and check for exactly what are the regulatory rules for this kind of adjuvant. For Leishmania, which kind of adjuvant you could use? Probably this one, QS21, which have been used, and that's probably the one you could try. Just one thing which is, I think it's quite important, and it was mentioned already before. A mouse is a mouse, a human is different from a mouse, and you can imagine that some adjuvants do not have the same effect in a dog and in human. That's clearly, and there are specific considerations for dogs, specific one for humans. For example, your cat doesn't like aspirin. Can be a problem for your cat if you give them aspirin. So that's the same. I mean, you have difference between humans and dogs. So Then protocol of immunization, quantity and number of doses, route and site of immunization. Do you want nasal, skin, and so on? Number of parasites for the challenge and the strain, the stage. Did you, at the beginning, do you want to use an ear model or foot pad model? And experimental you experiment when you do the infection. I remind you, as was said already, a needle is not a sandfly. So then you can go for your different phase. Phase 1. In phase 1, you do not really need the final process which will be used in clinical phase 3, but you need to define it already. Okay, it's the way I will go. The risk you have, it's not reproducible or scalable. I think you can. It's easy to immunize 10 mice. It can be a lot more difficult to immunize dogs already, or monkeys, or humans in terms of size, quantity. Then you have the problem of antigen potency or stability and the cost. Then you can go for a phase 2, which is a double-blind randomized. And probably the best way, since already some antigens do exist, I think if you can have an antigen in a specific formulation, you could use it as a benchmark and say better than. So you could use something which is known and see and compare to what was tested already. Again, with the same risk, not reproducible or scalable, and antigen potency and stability. So the key issues, I think we should get one day a Leishmania vaccine. It's known that we need a strong cell-mediated immune response, and we need experimental model. The problem is which animal model. Clearly, the mouse is a model, but I think that's the only thing you should do at the beginning and try to switch to monkeys or dogs. I think we need to test new adjuvants, at least use the one which do exist, like MPLSE, CPG, but then you can have all the problem with CPG. Testing new expression system, I think that's probably one way to go, as was mentioned, was using adenovirus or vaccinia virus or VLPs, and there are vaccines which do exist for hepatitis and papillomavirus using VLPs. That's probably another way to go. And the other thing which can be imagined or starting, it's live attenuated vaccine. They didn't work so well. And I think one of the problems is the presence of antibiotic-resistant genes that you have to remove afterwards. So it means you manipulate, you parasite more and more. Tarentulae, which was mentioned already, I think that's quite interesting, but you don't know yet how it will go into immunocompromised people. Can it be transmitted or not? And I think raise other type of questions. Then the release of GMO, that's another problem. I think maybe we should not care, maybe we should care. Do you have reversion possibility of something which has been attenuated or non-infectious to become infectious? And that's it. And so I'm on time. Try to make it shorter to be sure we can take the bus tonight.